Well, hello everyone. I wanted to give hopefully an interesting lecture that talks about some of the research I've been doing. And what I'm titling it is Bacterial Chemotaxis Control. I have a paper that gets into that's already published that you can look over. And what I'm trying to do now is look at a process comparison between three different bacteria. And so some of the resources I'm going to be using are these things that are listed up here. First is Uniprot, Ensemble Bacteria, and MIST, which is a microbial signal transduction database. And so I'll be drawing some information for each of these three bacteria and consider how that all works. So this is an illustration of where E. coli is in our digestive tract in our gut, and that'll be for a little bit of a reference. And also, one of the things that I'm leveraging in terms of the engineering side is something called the systems modeling language. And that'll be coming a little bit at the beginning and mostly at the end. And so here is a summary of the topics that I'll be going over. And so this is some work that I'm doing as we speak, and I'll get to it towards the end. And the material that I have here isn't as detailed enough in terms of the published information that I'm trying to leverage. And so I'm going to be moving to some other publications to help out. But the things that I'm going to show here are very informative and things that I've already leveraged in my paper that I published. So I want to give an introduction. So I'm doing that as we speak, but I'll give a little bit more context. Want to be giving you a perspective on what I was thinking about at the time as I was doing this research. What were the research questions and the themes? I'll then get into the bacteria comparisons of three bacteria. And then I'm trying to now think more as an engineer. And so, what is the functions that they have? Do they have similar functions? And then after that, I can get into the genome and the protein comparisons, look at the molecular biology side of things. And the focus of this research is getting into an area called chemoreceptors. And so I'll get a better sense of what that is as we progress. And finally, try and think out how could system engineering tools and approaches give us some greater insight. And then I'll give some synopsis of the findings. So here are the research questions. And so here is a first encounter with the three particular bacteria that I'm looking at, E. coli, E. meliloti, and R. sphaeroides. And I'm going to see, well, how do they compare? What functions do they share? How do the chemotaxis processes compare? And so the function is the general things that they accomplish. Now we get into this one area. How do they sense? How do they taste? How do they sense where the constituents that they're attracted to, that they want to move towards, and the constituents that they wanted to move away from? Those are things that we'll get a chance to be more focused here. And from a design pattern point of view, what do they leverage? And so, very common in software engineering and engineering, they have this idea of design patterns. And so, that's what I'm trying to introduce, that we think about leveraging that. What and at what level is the best basis for comparison? This is another area where trying to think about how to look at biological systems from an engineering point of view, where and how should we do it? And finally, how can the systems modeling language help? It's one tool. There's many things that are out there, but I'm going to be focusing on that. So the path, so what am I going to be leveraging as I get into this assessment? This is what I'd like to have us think about. So there's an engineering part, these two arrows on top, and then there's a biology part, these three down at the bottom. So engineering, we want to be taking things we're getting from the literature that characterize how research has categorized how a organism works. And so for this case, it's bacteria. System engineering is trying to think from a top-down perspective, and I'll introduce the methodology that I've been using in the paper that I published. And then the three aspects of biology. First, the molecular biology, look at the DNA side of things. What does that tell us? The microbiology, we're talking about an organism that's very small. And so what is the sense of being a bacteria? How do we leverage that and find an ecology? How does it actually work in a population of, say, E. coli, and even in that, a larger population, say, in the human gut? And so those are things that I want to give some perspective. And so, of course, this is how I would like it to happen, that it's a linear process. You just begin and you start to work forward. In reality, this is how it ends up being. Sometimes you even have to go back. And so it can be complicated. And so the nature of what I'm doing is multidisciplinary, and that makes it even more challenging. There's a rare audience that's going to be helping you be able to work through this material. So I wanted to underline that this is using an underlining engineering premise. If something is built and operational, then it must have been engineered. So that's what we're trying to use as a construct as we're looking at the nature of a part of bacterial function. So therefore, looking at the system characteristics will reveal how and why it is engineered in a certain way. And so if you haven't had a chance to review this paper, feel free to look at it. And it's called Bacterial Chemotaxis Control Process Analysis with SysML. And so this is published in the System Engineering Journal. There's various places that it's been referenced. I've just highlighted one of those. And so feel free to take advantage of that, and you can tell me what you think. So the method that I introduced in that paper is something called reverse engineering object-oriented system engineering method. Object-oriented system engineering method is already something that is published, but with this concept of reverse engineering, you have to get things into the right type of modality that you can do this object-oriented system engineering method. And so this is where a lot of this front-end work is required, surveying documentation, academic articles, textbooks, capturing the descriptive information that researchers and laboratory work has provided. And then you want to convert that to engineering prescriptive information where you have the concept of requirements for those that have experience of what it's like to be thinking in an engineering sense. And then when you have that, then we can be applying the object-oriented system engineering method, all the tools and resources that have been used to give some insight, analyzing architectures, design trades, and stuff like that. And so why not use a rich body of already existing resources? And so that's something I'm trying to allude to. So here is a high-level chemotaxis functional description. So some of you may not really be familiar with this, and so I'll be trying to also make it so people can have an understanding of that. And so there's basically these three parts. You detect certain substances, which are called ligands in the biology literature. And so we have these sensors, and so chemoreceptors, chemical receptors, if you will. And so these are the front end sensing. You process the inputs and adapt. That's things that happen in the middle. And there's some very small molecular sequences like key Y will be a key thing that will be part of this adaptation process that I want to give a perspective on. And then it responds with pre-programmed responses. And so say for a bacteria, if it finds a constituent, a substance that it's like, it wants to move in that direction. So there's a controlled response, move in the direction towards food, for example. So this was the first paper that I wanted to leverage in terms of this comparative analysis of bacteria by Armitage. And so these are the three different bacteria that they use. And so that's what I'm using at the moment for my efforts and research is E. coli, S. meliloti, and S. sphaeroides. And Judy Armitage is unfortunately since retired, but did some really wonderful work. And some of her colleagues have picked up and continued to develop it even further. And for the sake of what I'm going to be sharing in this talk, I'm going to be leveraging that, but I wanted to give you that basic introduction. So based on Armitage's effort, I'm going to be looking at these three different bacteria. And so in a very short form, and I'll give it from a couple of different points of view, this is basically what they do. And so E. coli in the human gut, it's in the intestine and it actually aids in digestion. I know there's good E. coli and bad E. coli. I'm not going to get into that. I'm just going to generalize it at this point and keep it in saying that it aids digestion. S. meliloti, it's in soil and it converts nitrogen. So it takes nitrogen in the air and translates it into N2 in the soil that plants can actually use. And so it's a symbiotic relationship between plants. And Sphaeroides, it's actually is in it can actually exist or not thrive, but it can exist in multiple types of soil. Moist soil, it can be in an oxygen-rich environment and then thrive, or in an oxygen-deprived environment, and it has more kind of resources, if you will. And so basically how I'm trying to summarize its top level function is to think that it's converting nutrients. So just using a knife example to try and compare these, I would argue that E. coli is relatively simple. S. meliloti is a little bit more complicated. So just going from a regular table knife to a serrated knife that is more specific function. And then Sphaeroides, I would argue it's kind of like a Swiss Army knife, that it has multiple functions. And so that gives us a nice spread of possibility. So this is a system modeling language diagram. And so what's called a use case, and use cases are meant to be very simple. And so if you were to talk with a stakeholder, this is how you'd be saying, did I capture the basic format of what you're trying to accomplish in stick figures and basic things? And so the noun would be bacteria, and then the verb is what we have in the use case. It supports an ecosystem. So this is a high level. The bacteria supports an ecosystem in an ecosystem environment. Well, let me just go one level below that and focus on what happens for these three bacteria that I'm looking at. First, for E. coli. And so the E. coli is operating in the human gut digestive system. And so the E. coli supports digestion in the microbiota. And that's a biology term about what is the nature of what E. coli is, one of various constituents that helps us to digest food that we eat. Okay, the next one, S. meliloti, it's in a different environment. It's mostly focused when it's in the soil around the roots of a plant. And so the S. meliloti operates in a soil ecosystem, and it converts atmospheric nitrogen into soil base N2. And this is the example of a nutrient that plants are going to thrive with, and so it's a wonderful symbiotic relationship. So you can see some similarities. They're very different in what they do in terms of the kind of ecosystem, but they're still related. It has this symbiotic relationship of helping something else, an altruistic type of motivation, if you will. Sphaeroides. And so here I'm pointing out to moist soil ecosystems. And so I'm just trying to capture it at a high level. And so I'm just saying that it cycles nutrients needed in moist soil. So it can do this conversion process like we saw for, in general, with a bacteria, a little bit more specific for E. coli. Talked about what S. meliloti does, and then finally R. Sphaeroides. Okay, now jumping into some aspects with molecular biology, and I would refer you back to the paper if you want to get a little bit of an introduction to some of these constituents that are used in chemotaxis. And so I'm not going to get into a lot of those details, but just mention that they're there. And so think of it this way. These are some key things. These first four boxes are part of a bacteria. So you have this front end sensing chemical sensors, and so chemoreceptors, and so they bond with these different types of constituents that they can are able to detect certain types of chemicals. And then there's two other, what I was stating are key elements, key A and key Y. Key A is more related to the sensing process. Key Y is more involved with signaling. And finally, the flagella. There is a distinction that the way that E. coli has many flagella, so this is a tail, while S. meliloti and R. sphaeroides only have one. And then I just give a rough summary of what type of ecosystems that they operate in. All right, the first one that I've gotten in more detail is talking about uniprot. And so how about looking at the proteins? And so I've mentioned key Y and key A. These are some key things I'm going to be looking at. And so using uniprot, they actually have characterized for these various types of bacteria these proteins. And so here is the uniprot name. And so I have it for E. coli, S. meliloti, and R. sphaeroides. And so there's four in R. sphaeroides, two in S. meliloti, and only one in E. coli. And the Armitage figure that I briefly showed earlier kind of gave you a sense of that. And so then looking for key Y, it's kind of going the same way, but you can see that now in the signal processing in the middle step for this bacteria, there's actually six versus there's four for R. sphaeroides. And so here we're getting a chance, a way of highlighting some of the relationship of function and with some of the key components. So here is just another bit of information of looking how large these proteins are. Some of them are larger than others, and the location of where they are in the genome. And I'll have a couple ways of looking at that in a little bit. And also for those that have some bacteria background, you can actually code in two different directions in DNA. For those that are on the engineering side, that's what I'm trying to highlight with these arrows, as you can see. So roughly speaking, the key A for the most part are about the same size. There is one that's larger, and so that may be some noteworthy things to be thinking about. And then for key Y, they're pretty close to being about the same size. And so just trying to look at the quote externals, taking some information what we can observe from these three bacteria. I'm going to get into more details of each of these bacteria one at a time. And the first one is the E. coli. And to give a basis, I use this perspective of thinking this as a regular table knife. And so it has some general characteristics, and so that's how I'm looking at it. And so, as I mentioned, what I'm focusing on is the E. coli that's operating in the human gut. And so this is just trying to illustrate what it is. And it has two modalities in which it operates with its multiple flagella, its multiple tails. And so they're actually located all over the bacteria. You can see a more realistic version here. And the chemoreceptors are all over the bacteria, but they can either come together when they're spinning in one direction, and so thus I can get movement in a certain direction, or otherwise they have a more of a random motion where they're like this. And so thus you can have like a pool cleaner that it goes across the whole pool in a random fashion, or an automated carpet cleaner that goes around your room and picks up dirt and what have you. So this is what we have for E. coli. This is the one that I'm going to focus on first. And I'll refer to some figures that are in the literature to give us some perspective of what is going on for E. coli. And so this was relatively speaking, a simple diagram, and so I was using that. And this is something that is from ResearchGate, and it just shows the chemotaxis pathway for the purposes of what I'm going to be focusing on, I'm going to be trying to keep it at a high level. And so just thinking of this chemoreceptors, that's kind of in this beginning part. The processing and adaptation is in the middle, and the response is at the end. So this is where we're getting the sensing with the chemosensors, the ligands bond with the chemoreceptors. Then there is a response. That's where, so a key component over here is key A. So that's why I have mentioned that. Key Y is a key element in the processing. And the phosphorylation is a more fancy word, and then it ends up giving it into control that you're going to be going in one of two ways, that you're either going to be intentionally going towards a concentration of food or away from a concentration of toxin, or you're just randomly going around the area that you're at. Okay, so um, now some diagrams from Forder, which is someone that worked with Armitage that I mentioned earlier, and he has a lot more detailed way of looking at information, and so I'm leveraging information for this talk and for this point in my research. And so he's looking at the E. coli chemotaxis pathway, and so there's 5 different chemoreceptors. And so this is trying to give you a little bit a sense of what that is and the response that you can have for each one of those constituents and what it might have. And so just like with this other figure that I showed you before, now I'm getting into more details for thinking about here. So we have the chemoreceptors, we have the processing, and then we have a motor. And so this is just an example of one of those motors. So now this is where I got into using one of the other 2 resources that I haven't mentioned yet. And so this is MIST. And so this is really helpful for giving information on the chemoreceptory systems. And so here we have, key Y is not part of this, but key A, key R, key B. You can see a little of that. Key B, key Y, those are part of that. Key A as well. So you can be seeing these things in various parts of these diagrams, depending on how detailed they are. And so I'm just going to be using this to give us a way of thinking about there's only one process that we have for E. coli. And so this is relatively speaking, as compared to the other ones, this is a simple. And so when I get into the other 2 bacteria, you'll get a better sense of what this means. And then another resource was the ensemble bacteria. And I'm not going to go into these diagrams in as much detail. I have more of an engineering background, and so I'm an engineer looking at biology. And so these are a huge amount of details, and I'm just trying to introduce those that say are on the engineering side, get some perspective. Those that are using these resources on a day-to-day basis will quickly be able to pick up some of these things. And so for E. coli, it only has 1 chromosome. You'll be seeing how the other ones that I'll be getting into details have more. And so then I'm going to be looking at these key components, key Y, key A. And so here is an example of, according to UniProt, the key A structure. And this may be one of the things I may have done a search incorrectly, but I was getting the same picture for key A as I was for key Y. But these 3D rendering of proteins can be useful for here is another resource that I looked at, EMBL, the European Bioinformatics Institute, and gave this a lot more complicated diagram for that. Anyway, moving on from that, that's just one of the things that I'm giving you the state of where I'm at in this. And so now we can also not only be looking at these chromosomes as a whole, well, we can dive into the specific area that we're interested in. So this is looking at for key A, and here is even a more getting in closer for that region. And so, and then blowing up that area is where you can be seeing this. It's large enough you can't see the individual nucleotides and stuff like that, the amino acids, but I just wanted to give you assessment. You can blow things up in as much detail as you would like. Okay, now we looked at key A. Now we're going to look at key Y. I already mentioned this before that this looked the same for key A, but just I'm going to let that go. This is going back to that other resource that gives us some insight. And so this type of rendering, there I'm just going to leave it at that. It may look like not everything is annotated or shown, but that's just another resource I just wanted to give in going into another resource that I'm just barely introducing, but it's not the primary three. So here for the ensemble bacteria, here is a top level view. There's only one chromosome for E. coli, and then going into more detail. And here, this is short enough that you can actually see some of the individual amino acids. And so this is a finer blow in. Turns out that key Y is shorter, and so we're able to do that. Here's a second bacteria, S. meliloti. And so I was using a perspective of thinking that maybe you could consider this a little bit more focused. And where does S. meliloti actually operate? And so this was from one of the papers that I referenced, and it was just showing how they were looking at cross-section of different colonies in one plant versus another, making some better observations rather than just using a single sample. And so the S. meliloti would be close to the roots, and they're converting the the nitrogen in the air and converting it to N2. And so having some air in the soil, it's able to do that process. And so thus we were thinking about, well, here are the things that S. meliloti does. And so there's 5 different type of chemoreceptors. I'll get to this later, but if you can think like an eye, the way that the human eye is designed, and so there's a spectral range that we can see in. And so let's just think of it from that perspective, that if this is 5 and this is larger, you can make the general argument from an engineering point of view that there's more things that it could sense. And so for staying within the context of what is important for this bacteria, it has more fidelity than, say, E. coli. But for E. coli, that you can make a counter argument that it doesn't need this extra sensing based on what it's meant to accomplish. This is adequate for it to be doing its intended function. Okay, what is the S. meliloti chemotaxis pathways? And unfortunately, this is where I ran into one of those roadblocks. I wasn't able to get enough information to be able to do this level of assessment like I was able to do with some of the other ones. What exactly are the chemoreceptors and how are they laid out and specified? I'm not going to get into this, but this was something I found is different. Psychometry. I don't even know how to pronounce it. It's not my area, but this was something that was related that I thought was at least noteworthy to put in this presentation. I'll just leave it at that. But back to the Armitage paper, this is how they laid it out. And so this was about as much detail as I could get. And so that's what we have. Okay, so S. meliloti, it has more capability than what we have in E. coli. And so we're going to be seeing some evidence of that when we look at it in these other types of databases. So this is MIST, the microbial signal transduction database. And so here are the various proteins that are used. and you can see that there's more individual types. And so 1, 2, 3, 4 types of things that are found there. So if I'm going to be focusing on key A, we can be looking at these different ones. And so there's going to be a rationale for having more than one, like we did in E. coli. And so this is what the genome looks for as meliloti. And so you have one chromosome, and then you have these two smaller things that are part of the way that things are laid out. So that's the general way of looking at it. Now, I'm going to go faster through these charts because there's more of them, especially when we get to the third bacteria for S. meliloti, key A1. So this is the information for that, getting into a top level view and then focusing down. I have all of that information for that. Key A2, once again, the top level information from UniProt, then getting into the ensemble database, looking specifically at key A2, and then drilling down to that. Key Y, first we have key Y1, and we have a top level, and then focusing down to more detail. Key Y2, pictorial, going down into the details, and we see that characterized. All right, so now we're into the third bacteria. And so this was an interesting one as I started to look at its capability and what the literature was commenting on in its function and its ability to adapt. And so basically it can actually be in an axonic, which is devoid of oxygen, so say a sludge type of environment, or it can be in an organic rich environment. And so it can actually switch back and forth between those modes and tolerate those types of environments. So here we can see, just looking at the chemoreceptors, we have 13. And so I would argue this indicates that it has a higher level of function. And so that's the reason why I use the analogy of thinking about a Swiss Army knife to consider this bacteria. And according to Porter, there's actually two different chemotaxis pathways that he thought were noteworthy. And unfortunately, we only have limited information about the type of chemoreceptors that are talked about specifically in literature for this one in this article, and I'm listing in there. So this is the first pathway. And here is the second pathway. And so Porter does a good job of expressing this. And you can see there is a difference between them. This pathway 1 is more complicated. Pathway 2 is a little bit more streamlined. And then I have the same kind of information that I expressed before. And so this is for the chemosensory systems. And so key A, that's the one I've been mostly focused on to characterize this. Key R, key B, and key W, which I talk a little bit about in the paper that I published, but I'm just focusing mostly on the key A and the key Y between these three bacteria. And you can see how the genome for this bacteria is more complicated. It has 2 chromosomes and these 5 other small parts that it has some of its genetic information that's in those areas. Once again, I'm going to have to go even faster, but it just was to characterize this information. So key A1 for sphaeroides, the picture, high level of that, and then getting into the details. Key A2, picture, high level perspective, getting into some more of the details from ensemble bacteria. Key A3, a picture, 3D rendering of a model, I should say. Then we get into the chromosome where key A is, and so this is A3, and getting into more detail. Key A4, same things. And then we have the key Y, and so we have the key Y1. You can see the top level rendering of how the 3D structure is envisioned to look like. The chromosome that contains it, getting into that information gradually getting into more detail. Key Y2, same thing. Key 3, key Y3, key Y4, key Y5, key Y6. So that's a lot of information, and you can see, so if you're on the biology side, this is something that it's quite interesting and something that may be closer for you to be handling day to day. But for engineers, this is where it comes back to that reverse engineering, open object-oriented system engineering method. How do we take that into a digestible fashion to do a top-down assessment of what we're seeing? So this is where I started to why I chose certain things. I looked at key A, which is representative of what's going on in the chemoreceptors, key Y, which is looking at the processing of information, and also included the number of flagella. This isn't really the focus of what I'm trying to do with my chemotaxis analysis, but I wanted to make sure that I include some thoughts on that. So I already showed you this chart before. So now I'm getting into thinking more as an engineer, and I'm going to be bringing in some SysML diagrams again. So we can be looking at here is the top level function of what they do. So E. coli, they're aiding digestion in the gut. S. meliloti in soil, it's converting nitrogen to a form that can be used by plants. R. sphaeroides, it's in moist soil and it's converting nutrients. And I mentioned that here is the high-level functional way of looking at what we're accomplishing. And I'm going to be talking a little bit about each of these processes. And so we'll keep walking through this chart. So key A was key for this first part, trying to be thinking about the chemoreceptors. And now introducing a block definition diagram in SysML. This is how I was trying to be thinking of these three different bacteria. And I'm using the word forms because unfortunately it turns out that the actual molecular composition of the proteins is not the same. So is it appropriate to consider that these forms can be categorized in a similar fashion? I'll come back to that. So E. coli, there say is one that's across all three of them. And then another form of key A is in R. sphaeroides. And so maybe that's one way of trying to wrap that up to a higher level. So here is the types of chemoreceptors. And so once again, I'm dealing with this front end here. And so I'm trying to see what's available for the literature for these three different bacteria. And unfortunately, there's a gap that I'm missing in a couple places. And for the chemoreceptors, the R. sphaeroides is one that I don't have all of them. And so I have not been able to find it. Maybe someone else has. You can always feel free to disseminate that information if you like. So that's some additional information from the chemoreceptors point of view, thinking of from an engineering sense. Now thinking about the processing and adaptation, focusing on key Y is how I would try to represent that. And we see that R. sphaeroides has 6 different forms. And so I'm trying to be thinking that same way. So we have some similar things. S. meliloti actually has 2 key Ys. So you can see that the processing is more complicated in the S. meliloti and R. sphaeroides. Even more than E. coli. E. coli only had 1 key Y and only 1 key A. So that maybe gives us a perspective of how we can be relating those different types of functions and capturing it like that. Finally, I included the flagella, the tails, and it turns out there's another way that you could be annotating that in a SysML block definition diagram. And so you can have this dot, dot, and asterisk means there's can be multiple flagella, but the other ones only have one. So is it fair? Is it good to be considering these flagella all the same? Having to navigate within soil, I could make a argument that maybe I have to have a couple layers of definition that this flagella may be a more powerful flagella. And since there's multiple flagella, you could argue that each one of these flagella are individually can be more not so robust, but those are some details I can just leave out there and have you consider. Well, what about the protein and sequence comparison? I was trying to look at a way to compare the three bacteria. There are some functional similarities, but the key Y sequences between the bacteria are not the same, and the key A sequences between the bacteria are not the same. So what do we do with that? Is the same is so do they have the same function, but there's different implementations? And just to give you an analogy, like you can think the way that a bee versus a bird versus a bat, how they fly, that they all fly, but the implementation is different. So maybe that's one of the another layer of nuance that needs to be captured, since we don't see that they're the same. So they're not the same. I'm just going to have a few slides that illustrate that to a degree. And this can get a little bit complicated, but I'm just going to try and keep this a high level. So I'm leveraging information from UniProt. So all three of these different bacteria, E. coli, S. meliloti, and R. sphaeroides. And so here are the specific proteins that they have, some information that's captured in the UniProt database. And then I get into the sequences, looking at the amino acids. And so key Y is short enough that I could actually show this. It's still getting a little bit challenging in the slide, but you can get a chance to be seeing the whole sequence. I then blew it up so you can be seeing the first part, and they don't match. There is maybe some correlation, like you can see down here. There is some relatedness to the sequence, and maybe a little bit between the S. meliloti and R. sphaeroides, but that's something I've just looked at a little bit so far. Here now is the key A protein. These are longer, and so it's going to be a little more challenging, but I've listed the general information, the name of the proteins, and then you can see that this is probably totally not readable, but these are longer, so I was trying to capture the whole information for them, and then just had the first 152 amino acids to give some comparison. And we can see that these are fairly different. And so that was helping me to be thinking about them more in this analogy of using flight as an example, but there's three different mechanisms for how flight is accomplished. So for the engineers, in case you're not familiar with the amino acids, and they have like a single letter that's used to summarize what those are. And so you can see here, all of these letters correspond to one of these. So just give you an example, M, that you can see the information for that. Okay, one of the things I tried using some graph theory and number theory, I was trying to see if there's maybe another form of metadata that might help us to be thinking about this. And so this was trying to look at a key A comparison. And so what this does, all it does is count the number of each type of amino acid for each of these bacteria. And so this is the totality of the key A that we have for E. coli, S. meliloti, and R. sphaeroides. Maybe there's some similarity for that. I'm still playing with that. Then for the key Y, there's more of them. Once again, maybe you can see some similarities between those. Is there enough distinction between what we see in key Y and key A to draw some useful metadata information? I'm still trying to be thinking about that. And finally, this is trying to hook all them all together. And maybe you can see a little bit of a difference between the key Y and the key A. I'm hoping it will be something that's useful, but I'm just including it here for discussional purposes. Okay, let me just finish up with a few charts here. So questions from the finding, although the genetic sequences differ, do the chemotaxis proteins perform similar functions in one bacteria type to another? And so that's still something I'm looking into. And I think there is, but the how to be breaking that down is something I'm still working on. Is there a form of quote metadata that can help? And I was trying to think of these spider plots as a way of maybe seeing if that can be a helpful modality. Are chemotaxis proteins the best level to consider as modular functional units for the reverse engineering, object-oriented system engineering method exploration? And so I'm thinking about that, and we'll continue to go forward with that. So here are the way that I did my assessment so far. I looked at key proteins, key A for the chemoreceptors, the key Y for the processing, and I also annotated the number of chemoreceptors. And so key A gets into the chemoreceptive signal processing. The chemoreceptors are what chemicals are it actually able to sense. So here is a way of trying to make it more understandable for the engineers and maybe more complicated for those biologists that are looking at it. So here is an analogy for an engineering analogy. So you detect certain substances or ligands, so detection. So you can be thinking of a camera, and it's able to have certain sensings. You have color, digital photography, and so basically it would be similar to be thinking of different types of bands that you have. And so a red, blue, green, for example, you have these different technologies. CCD or CMOS. And so I was using key A to give some sense of that. For processing and adaptation, automated gain control. So changing the volume. And so say if you're in a loud environment, you start to lower the gain. If you're in a quiet environment, you start to increase the gain. There's various types of electronics that do that. And so this just gives you an interesting way of thinking about adaptation. You also need to translate the information and relay the signal. So you have to have a code, and then you have to have a mechanism for the channel to have the information pass from point A to point B. And so I discussed that in the paper that I showed earlier. And finally, there's the response. And so you can be thinking of having actuators and a propeller. And so the rate of speed in which it is driven is based on what it's sensing, what type of result that it computes, and says that I'm going to be going in a certain direction and how fast. So those are the things that I wanted to give as a perspective. And then as a summary, bacteria provides a beneficial supportive role in various environments, an altruistic helper, which is kind of interesting, at least for the bacteria that I'm focusing on. We want to seek to fill in the missing biological inputs. One of the ways I'm dealing with that is I found another resource that gives all of the chemoreceptors, and so I may be focusing on a different set of bacteria to do this comparison. What I'm trying to do is look at the bacteria from an engineering point of view and doing an analysis, a comparison of designs, if you will, or engineering architectures, using things that are used in the engineering domain to help us. And so I'm also exploring the implementation nature of the function commonality. And so a functional connection at a protein class. And so I was looking at these two different proteins, key A and key Y in particular, and then think about the details of the molecular biology's relationship to function in the work that it performs. I've showed a couple examples in how you can be utilizing the systems modeling language to explore this, and that's something that I would like to explore further. Okay, thank you very much.